Cell membranes. All right. So the function of cell membranes is to form boundaries between the inside of the cell and the outside of the cell. It's also going to function to regulate movement into and out of the cell, meaning bring stuff into the cell, bring stuff out of the cell. Now, cell membranes follow a fluid mosaic model. Fluid meaning it's constantly interchanging parts. The phospholipids that compose it are constantly being exchanged between one another, flowing from side to side and in between. And then it's a mosaic because it's actually made out of many components. So it's dynamic, referring to the fluid part. So it's a dynamic membrane with many lipids, proteins, carbohydrates embedded. So that's the mosaic part. Lipids are going to provide the hydrophobic barrier to water-soluble molecules, meaning water-soluble molecules are going to be polar. Polar substance, large polar molecules, for example, have a lot of trouble actually making it through the phospholipid bilayer. So the lipids themselves, that's what's actually preventing them from those larger polar substances from moving through a membrane. Now proteins, the protein component of your cell membrane, those are going to constitute your channels or transports, so your transport proteins. You're also going to have carbohydrates that are embedded, and sometimes glycoproteins or carbohydrates bound to proteins, and those could serve as recognition sites or cell signaling molecules. Now, when it comes to the cell membrane, it's going to be composed of something called phospholipids. So here is a single phospholipid, and when compared to a triglyceride, for example, a triglyceride has 3 fatty acid tails and a glycerol backbone. In the case of a phospholipid, you actually have a choline head, which is polar, and then instead of 3 fatty acid tails, you only have 2. So you have 2 fatty acid tails and then a choline head, which is polar, making this actually an amphipathic molecule. So we have phospholipid here, phospholipid here, phospholipid here, phospholipid here. And then if you look at the other side of it, you'll notice that it forms a bilayer. So you actually have 2 layers of phospholipids that compose your cell membrane. So mostly phospholipids. Now it's going to have that hydrophilic or polar head. Like dissolves like, so polar stuff likes polar stuff. You're going to have water on the outside, you're going to have water on the inside of your cell, and so those heads that are polar are going to face those polar areas, or areas that have a lot of polar stuff like water. Now it likes water, so we consider it to be hydrophilic. Philic meaning lover of, hydro referring to water. So those heads face water or polar substance, and then it has a hydrophobic tail. So you have these non-polar tails as a general rule of thumb. When you see long carbon, hydrogen, or hydrocarbons, carbon-hydrogen chains, typically you can associate that with being, or you could assume that that's going to have non-polar characteristics. So these fatty acid tails are going to be non-polar, and if they're non-polar, they're not going to like polar stuff such as water. So therefore, they're going to be hydrophobic, afraid of water. So your phospholipids are amphipathic. Part of it faces the outside polar region, and then the other stuff, the non-polar stuff, faces itself in this larger non-polar region. Now, usually larger polar molecules have trouble getting in through the membrane because the vast majority of your phospholipid bilayer is actually going to be non-polar. And that polar stuff, well, it's going to want to hang out with polar stuff such as the phospholipid heads, and it's just therefore going to stay on the outside of the cell or stay on the inside of the cell and not really transport through the membrane on its own. And then, as we've already kind of discussed, it's a bilayer, so those tails are going to face each other with the heads on the outside, and movement within the membrane is easy between those phospholipids. Now, your membrane fluidity, remember all of those phospholipids are going to be moving in between and exchanging with each other. The fluidity actually will decrease the more cholesterol you have deposited within that phospholipid bilayer. So cholesterol is going to add to its rigidity, or the cell membrane's rigidity. If you have a lot of saturated fatty acids, as you might know, your saturated fatty acids are going to have no double bonds on the fatty acid tails, whereas an unsaturated fatty acid tail has at least one double bond, a monounsaturated fatty acid. And by saturated, we could actually just say that saturated fats are saturated with hydrogens. And unsaturated, well, it's less saturated with hydrogens, because if you form a double bond between two carbons, then you have to yield one of these carbons bonds. What does it yield? It's actually yielding one of those bonds that would otherwise have with a hydrogen, so it's less saturated with hydrogens. These unsaturated fats tend to be a lot more flexible, jiggly, and if you have those embedded into your membrane, unsaturated fats, you're going to have more fluid, a more fluid membrane. However, when you have more saturated fats embedded in your phospholipid bilayer, it's going to be, therefore, a little bit more rigid. And then lastly, your fluidity is going to also decrease with decreased temperature. As we know, as a general rule of thumb, higher temperatures would probably speed up reactions. In this case, higher temperature would probably increase the rate at which your phospholipids exchange with one another within that fluid model. Decreasing the temperature is going to lower the speed at which they exchange between one another. So let's talk about the proteins that we're going to find within our phospholipid bilayers. So they can be both hydrophilic and hydrophobic, meaning they can have polar regions or they could have nonpolar regions. So if you look here at this transmembrane protein, it's got a nonpolar region, so domains with hydrophobic surfaces, and then the purple is domains with polar surfaces. So the nonpolar is going to want to hang out around nonpolar stuff, your phospholipid or the fatty acid tails. And then its polar regions is going to want to hang out with polar stuff. You got water on the outside here, you got water on the inside here. So this is a transmembrane protein because it traverses the entirety of the membrane. Now you could also have proteins that are embedded but don't traverse through the entire membrane. This would just be a integral membrane protein. So it's amphipathic with both polar and nonpolar parts, but it's just kind of like embedded into the protein, into the phospholipid bilayer. And then you have peripheral membrane proteins. Those are just going to latch onto those polar heads of your phospholipid bilayer. So not embedded necessarily on the inside or outside of the membrane. They're just kind of latching to the surface of your membrane. So these proteins, again, it's a fluid mosaic meaning it's composed of many parts, and it's fluid meaning all of the parts are going to be constantly moving among one another. And so these are going to move freely among the phospholipids. All right, lastly, let's talk about the carbohydrates. So glycolipids, glyco meaning having to do with sugars, lipid meaning having to do with some kind of fatty acid. A glycolipid is a combination of both a sugar and a fatty acid. So here would be a glycolipid bound, it's a carbohydrate chain bound to some kind of fatty acid. Now you have glycoproteins, sugar, protein, and that's just going to be a carbohydrate bound to a protein. So typically your glycoproteins are going to function in cell recognition as well as adhesion. So we'll talk about later in this lecture about adhesion. Your glycoproteins are going to be involved in latching cells to one another, and again, also going to be involved in cell recognition. So on the surface of all of your red blood cells, for example, you've got glycoproteins of a specific type that the rest of your friendly cells use to identify one another and prevents them from eating one another or identifying one another as enemies or invaders.